Hey everybody. Today we're gonna talk about action potentials and neuronal function, but more specifically, we're really gonna focus on an integrative kind of look at how all of the things we've talked about, including graded potentials and excitatory postsynaptic potentials, all of this stuff, how it's all kind of connected to one another, and what it kind of looks like in a sort of schematic that is depicted this way, okay? So, and then, at the end, we're going to correlate it a lot with this action potential graph, okay? Now, again, obviously, this is not going to cover everything. This is a specific scenario that we're going to look at, specifically of a sympathetic scenario, sympathetic nervous system. But it is a great review resource, and I highly recommend this, especially for introductory courses. So, without further ado, let's get started, okay? And if you guys like this video, make sure, please like the video and subscribe for more. We have other videos on this. So, let's get started. So, first of all, we're going to take a look at the quick anatomy of the nervous system, right? So here you have your spinal cord in pink, okay? And you have the sympathetic chain ganglia in yellow, okay? And that's what we have there. Okay, great. So why does that matter to me? Let's kind of, let me erase this. Let's take a look at what this kind of looks like. So inside of the spinal cord, okay, there is this neuron. Okay, and this neuron is going to synapse, okay, it's going to leave the spinal cord, and it's going to synapse with another neuron that is residing in the chain ganglia, okay, in the chain ganglia. So this is one type of ganglia in the sympathetic nervous system. We have other types, like we have collateral ganglia, and we have that special scenario with the adrenal medulla. But for the purpose of this video, we're only going to cover this specific kind of ganglion, but just remember, there are other types of ganglia for the sympathetic nervous system. And of course, when we're talking about the parasympathetic system, we have the terminal ganglia. So, that is that. Now, let's kind of zoom in. Let's take this guy and let's zoom in on it in this big picture. And let's see what this neuron kind of looks like, okay? And what are its functional properties, okay? So, okay, so we got two neurons here, okay? It's the exact same two neurons that we were looking at before, but you can see they look a little bit different. One is a bit myelinated, okay, so it's myelin, and the other one has no myelin, no myelin. You guys can remember that myelin, when the myelin is present, it increases conduction velocity, one, and two, it's actually going to lower the presence of voltage-gated sodium and potassium channels, okay? Which we're going to mention, okay? But you can kind of already see it, okay? So these channels, there's less of them here in the myelinated as compared to the unmyelinated group, okay? All right. So now let's kind of start digging into things. But firstly, I got to mention this is a preganglionic neuron, and this is a postganglionic sympathetic neuron, right? So preganglionic sympathetic neuron and postganglionic sympathetic neuron, okay? That's important to understand the difference. Now, here is another thing. Where's this ganglia? Right here, okay? This right here is a ganglia. This is a ganglia, okay? This yellow thing is a ganglia. It's supposed to represent the sympathetic trunk that we saw right here, okay? So that's the ganglia. Great. So this is the ganglia. So let's kind of zoom in back over here and see what everything looks like. So how is an action potential generated, guys? Right? What needs to happen? Something needs to happen to generate an action potential, okay? And, for more often than not, it's going to be the opening of some sort of ligand-gated channel, which leads to either an EPSP or an IPSP. And, that's going to either depolarize or hyperpolarize a cell. If you depolarize it enough and you reach the specific threshold required, we're going to launch an action potential, okay? So just a quick review for you. Now, let's say we have two excitatory neurons here. Okay, this is an excitatory neuron, this is another excitatory neuron, okay? So we know that. So again, this guy's excitatory and this guy's excitatory. Let's say only this guy launches an action potential down its axon and it releases certain chemicals, okay? And these chemicals are going to be called neurotransmitters. So let's zoom in on this guy. Boom, 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 boom. He releases a whole bunch of neurotransmitters, okay? One of them will bind to this pocket, okay? This receptor pocket. When it binds to this, okay, so what is this guy? This is a neurotransmitter. I'm going to abbreviate it as NT because I'm a little bit lazy. But when it binds to this pocket, something cool is going to happen, guys. This is a gate, okay? It's a ligand-gated channel. This gate will open up. When this gate opens up, okay, something cool is going to start flowing in here. Okay, remember I told you this is an excitatory neuron, right? So if it's an excitatory neuron, what kind of ion would you expect to start flowing in to the cell? That's right, it's going to be sodium. Okay, and let's make this a little bit less thick. Okay, so it's going to be our good friend sodium. He's going to start coming into the cell. Okay, boom, boom, boom. He's going to bring positive charges in here. Okay, he's going to make the cell a little bit more depolarized than it was before. Because remember, the resting memory potential of a neuron is give or take negative 70 millivolts. And by opening this gated channel, this guy brings in positive charges to try and depolarize the cell. Okay, so that's that. But on the surface of every cell in your body, there is this special transporter, okay? What does this transporter do? This transporter takes 3 sodium ions and pumps them out of the cell, okay? But it also takes 2 potassium ions and brings them into the cell. What is this guy called? This is your sodium potassium ATPase. I'm not going to write that down. Okay, but this is your sodium potassium ATPase. Why ATPase? Because it utilizes ATP to pump these guys against their concentration gradient. And this guy helps with maintaining ion concentration gradients, as well as it contributes a little bit to the resting memory potential, about 20%. But the majority, the 80% of it, come from the leaky channels. Either way, we opened up this ligand-gated channel by an excitatory neurotransmitter binding to it. A lot of sodium started to rush into the cell, okay, and depolarized it. But did this cell reach threshold? Here's the question. We're going to say no, it didn't. Why? Because this is a single neuron launching an action potential on a postsynaptic neuron, right? So this would be a presynaptic, and this guy in this scenario would now be a postsynaptic, right? You guys agree with me here? Hopefully you do. Now, if that's the case, one neuron acting on another neuron to stimulate it, what is that called? What is that called? It's called temporal summation, right? So this is temporal summation, okay? Temporal summation is rarely going to get you to threshold, okay? All right, so now let's take a look at something else. Okay, let's take a look at something else. Let's say, oh, and by the way, what happens to these positive charges that come in? These positive charges, guys, because they didn't reach threshold, they didn't make the cell reach threshold and launch an action potential, they're going to actually get put back outside of the cell by the sodium potassium ATPase, okay? So that's what's going to happen to these positive charges. Okay, so that's that. So let's take another scenario. Let's say we still have this excitatory neuron launching an action potential here, okay, and opening up this ligand-gated channel and allowing for sodium to come in. But at the same time, there's another neuron right here who is doing that exact same thing and is opening another ligand-gated channel. Okay, he's opening up another ligand-gated channel. If he does that, again, this is another excitatory neuron, sodium is going to start rushing in again. And when sodium rushes in, okay, remember now, both of these are happening at the same time. So both this and this, they're both happening at the same time. If this occurs, this will be enough of a stimulus to get this cell to threshold potential. What is the threshold potential? What is the voltage, guys? I know you know it. It's negative 55, give or take. Okay, it's about 15 millivolt difference off of the resting membrane potential. So, negative 55 millivolts. Okay, but before we talk about what this threshold potential is going to do for us, I first got to mention something. Initially, we said temporal summation was not going to be enough to get us the threshold, right? So what do we have to do? We're going to have to do what's called a temporal and spatial summation. Okay? A temporal and spatial summation, you guys already should know. Spatial summation is when multiple neurons act on a postsynaptic neuron, okay? So here is one neuron and here's another neuron, and both of these guys are acting on this postsynaptic neuron, okay? But this guy was originally acting as a temporal summation. So we kind of did a combination of temporal and spatial summation, okay? They actually give this a name. They give this a name. They call this spatial temporal summation. And usually this is the kind of summation you need to launch an action potential. Okay? This is the kind of summation that you usually need. Now, quick little recap. We did spatial temporal summation, right, which released these excitatory neurotransmitters, which bound to the ligand-gated channels, okay, ion channels. By the way, which kind of channels are these guys? They're ionotropic channels. Why ionotropic? Because they directly allow the passage of ions through themselves. Okay? So they're ionotropic channels, and we opened them, and we allowed sodium to come in. Once sodium comes in, okay, a lot of it starts to come in, and a lot of that kind of voltage starts to summate on top of another, okay, which makes the cell reach threshold potential. Once you reach threshold potential, you guys should already know we launch an action potential. But how? Here's how. Boom. Boom. These channels get stimulated. What are these channels? What are these special channels? What are these darn channels? These channels are going to be your sodium voltage-gated channel and your potassium voltage-gated channel, okay? If you recall, I haven't drawn the structure here because it'll make the drawing a little bit too complicated. But if I bring you up here, you should recall the structure, okay? Here's the sodium gate. So here's the sodium voltage-gated channel. And here is the potassium voltage-gated channel. Potassium voltage-gated channel, okay? This guy, okay, had a specific morphology to it. He had an activation gate, okay, which opened slowly and closed slowly, okay? This guy had an inactivation gate and a activation gate, okay? Which, by the way, this guy snaps open at threshold potential, and this guy starts to close slowly at threshold potential. And you guys will see later on when we go through the graph, you know, how long it takes and where it's closed and all that stuff, okay? But just wanted to give you guys a little bit of a reminder as to what is what, okay, and their morphology. So let's go back to our drawing here. So we managed to generate enough of a voltage to hit threshold potential. What are these positive waves called? Okay, so you know these are a graded potential, right? This was a graded potential. What kind of graded potential was this? This, my friends, was the excitatory postsynaptic potential, okay, that kind of built up all over and allowed for this threshold to be reached, okay? So let's say if the cell was at negative 70, we brought it to about negative 60 with this first wave of depolarization, and this second wave really brought us to about negative 55, okay? All right, just making sure you understand that. Let me get rid of all this extra drawing, okay. So, what does this threshold do? We already said it's gonna open up these channels, okay? So what's that gonna look like? When I open up the sodium voltage-gated channel, sodium is gonna start rushing into the cell, It's going to start rushing into the cell. And what is this sodium here going to do? It's going to allow for this channel, this next sodium voltage gated channel, to become open, okay? Because it's changing the voltage in this area, okay? This is the new active zone, if you will call it, okay? So it's going to open this guy. And some more sodium comes in, and same thing, okay? This guy will come in, and he'll do the exact same thing. But at the same time that this is happening, guys, something else is also happening. Check it out. At the same time this is happening, these guys, these potassium-gated channels were getting stimulated. So potassium is going to leave, potassium is going to leave, and potassium is going to leave. But you might be thinking, well, the threshold potential activated this channel, right? So it can open. How do these guys activate? What's stimulating the change in voltage? Great question, my friends. It is these sodiums that come in. These sodiums are going to initiate the activation or the opening of these channels, okay? And so again, you can put an arrow here that sodium is doing that, okay? All right, so sodium, because of its voltage that it's coming in, it's going to open up these channels and, you know, make it rush out, okay? All right, so again, we can show potassium going out, and we can show sodium coming in. Now, here's the thing. Just a quick note about the morphology I want to make here. Notice the amount of channels I put here, and notice the amount of channels on the rest of the axon. On this portion of the axon, okay, there is a higher concentration of channels than over here. Why is this a scenario? Why is this the case? Because this is a very important portion of the neuron. It's called the axon hillock. The axon hillock has the highest concentration of voltage-gated channels, and that helps with the propagation of the action potential along the nerve membrane, okay? All right, so I just wanted to make sure you guys understand where the axon hillock is. But either way, this wave of depolarization will continue down the length of this neuron, and the wave of repolarization is falling right next to it, by the way. I should mention that. Okay, but you guys should already know that. And you guys remember that this cell is myelinated. It is myelinated. And so because of this, you can see these charges that are coming in, these positive charges, they're going to travel a longer distance rather than having to stop in the middle and open another channel. Stop in the middle and open another channel, okay? So they're traveling a longer distance in a faster time. So they come here, they open this guy up, they open this guy up, boom, great. Sodium will start to come in again, and potassium will start to go out. It'll rush out, okay? Great. And this is kind of like your action potential, okay? Compare this to the graded potential that we were talking about in the start. Remember, it wasn't great enough, so it got taken back out by the sodium-potassium ATPase. But the action potential is so strong that it can regenerate itself. It's not decremental. That's what we call it, okay? It's not decremental. Now, what happens to all these positive charges that came in, okay? So we got a lot of positive charges that came in, okay? Where are they going to go? They're going to go to two places. So they're going to come here, and they're going to come here. They're going to open up these channels, these specific channels, okay? These channels, what are they sensitive to? How do they open? If you said voltage, that would be correct. And if you're confused on why, the answer is these positive charges, right, they give a voltage to the cell, right? We agree here, right? They bring the cell here to about positive 30, let's say. Okay? So if they do that, look what's going to happen. They're going to come here and they're going to open up this calcium voltage-gated channel. Calcium voltage-gated channel, okay? And it opens up at about negative 40, okay? At about negative 40, if you're curious. But here's the calcium voltage-gated channel. Okay, voltage-gated channel. Okay, and the calcium is going to start coming into the cell. Okay, so I'll draw it here as well. Calcium will start coming into the cell. Why does that matter? Why is that important? Why is it important that calcium comes in? Because you see this here? What is this, guys? This right here is a synaptic vesicle, and these red dots in it, they're going to be your neurotransmitters. The neurotransmitters are inside of synaptic vesicles. Okay, cool. What does calcium do with all this? Well, let me tell you. On the surface of this synaptic vesicle, there's a special protein that's called synaptotagmin. Okay? And this synaptotagmin, it senses intracellular calcium, okay? And once it senses that calcium, okay, it'll actually go ahead and bind to this protein here, the snare protein. You can see how they kind of have like little ends that would match each other, right? So it'll go and bind to that thing, and it'll allow for the exocytosis of its contents, right? So it'll release the neurotransmitters into, what is this area called? This is the synaptic cleft, okay? Synaptic cleft. It's about 10 to 20 nanometers in length. 10 to 20 nanometers, give or take. Now, that's what's going to happen, guys, okay? That's what's going to happen. Again, I'm going to erase this just to make it look pretty. So, but this is synaptotagmin. It senses this intracellular calcium, okay? And it's going to bind towards its snare protein. Okay, this is a snare protein, guys. Snare protein, okay? Or, docking marker receptor, whichever you want to call it. It'll bind to that. Now, again, this is extremely simplified because there's other proteins involved here. There's synaptobrevin, there's syntaxin, SNAP25, all these other things, right? But this is just super, super duper oversimplified. Right. So just a quick review of what we've been talking about so far, okay? We started off with temporal summation, which was not enough. It wasn't enough to stimulate this neuron to reach action potential. But then we did what's known as a spatial temporal summation, or you can call it temporal spatial summation, but this is a more common used word, okay? And together we opened up a whole bunch of these ligand-gated ionotropic channels, okay, which allowed for sodium to come in and depolarize the cell to the point where we reach threshold potential. This threshold potential initiated an action potential at the axon hillock, which continues to regenerate itself down the length of the axon, okay? And in this case, because we have a myelinated neuron or axon, there are less concentrations of voltage-gated channels, so the positive charges can just skip or jump over, not actually, but jump over to the next channel pretty quickly, okay? Allowing for a faster conduction velocity. Right, so once these positive charges finish going down the length of the axon, they're going to stop over here, and they're going to activate these voltage-gated calcium channels, which again open up at about -40, right? Remember these two? They all open up at -55. This one opens up at about -40. And he's going to let a whole bunch of calcium come into the cell. Again, same thing on this side. A whole bunch of calcium comes out, okay? And when all of this calcium comes out, this special protein on the synaptic vesicle called synaptotagmin is going to sense that calcium, okay, that intracellular calcium, and he's going to allow for the binding of synaptotagmin to the snare proteins, which causes the release of its contents, exocytosis, as you guys should know that, okay? So what does this guy release? Remember, don't get caught up by all of this stuff here, okay? We were talking about a scenario where we had a post-ganglionic neuron, sympathetic post-ganglionic neuron interacting with a sympathetic, let me repeat myself. We had a scenario where a sympathetic preganglionic neuron was interacting with a sympathetic post-ganglionic neuron, okay? So if they're part of the sympathetic nervous system, right, what is this neurotransmitter here going to be? If you said it's going to be acetylcholine, then pat yourself on the back because it is indeed acetylcholine, okay? This is acetylcholine, and acetylcholine is going to bind to a specific receptor here, okay, this guy, okay? What is the name of this guy? It's often written like this, NACHR. N stands for nicotinic acetylcholine receptor. So the nicotinic acetylcholine receptor. If you're wondering why it's called nicotinic, it's because it was discovered using nicotine. Okay? What kind of channel is this, guys? This is also an ionotropic channel. Ionotropic channel. Okay? So this channel is going to be specific for the neurotransmitter acetylcholine. Okay? And he's going to bind acetylcholine. When he binds acetylcholine, its gate, remember it's a ligand-gated channel, so its gate is going to open up, okay, and allow for what to come in, guys? Our good old friend that we know so well, sodium. He is going to come in here, okay, and he's going to start depolarizing the heck out of this cell, okay, inside of this actual ganglia. This is a ganglia here, okay, sympathetic chain ganglia specifically. All right, so he's going to depolarize the heck out of the cell until what happens? Until it reaches what? Until it reaches threshold potential, negative 55 millivolts. Once it does that, what's going to happen? That's going to stimulate the opening of the sodium and potassium voltage-gated channels, which, like you remember from this previous example, it's going to do the exact same thing, right? Again, at the axon hillock. Axon hillock. Okay? Same thing, guys. So once it does that, again, sodium will come in, it'll activate the next channel, okay? But at the same time, it'll induce the opening of this channel, okay? Where potassium can go out. Oh, sorry, not this channel. This channel. And again, potassium will go out. Potassium will go out. Positive charge activates here. Boom. Boom, right? It's the same thing, guys. I'm not going to keep doing this, but it's the exact same thing as the last scenario. The only difference here is there is no myelin. If there's no myelin, the conduction velocity is going to be much slower in the postganglionic neuron. All right, so look, he's going to come here, activate this guy, positive charges come in. Come here, activate this guy, positive charges come in. Make the cell more depolarized, boom, boom, boom. Again, same thing. Same thing as before. It's the same thing as before. And again, meanwhile, potassium is also leaking out. That is repolarizing this section and actually making it hyperpolarized as well. And then repolarizing this section and then repolarizing this section. Okay? All right. So, great. That's great. Now, again, same thing. We're going to come here. These positive charges. Look at these guys. They're going to come over here and they're going to stimulate the opening of this channel. What kind of channel is this, guys? This is a calcium voltage-gated channel, and the calcium will come in, okay, and it'll actually stimulate synaptotagmin, right, this molecule here, synaptotagmin, and then synaptotagmin is going to bind to these snare proteins, causing the release or exocytosis of these neurotransmitters, okay? These are neurotransmitters. Now, one thing I got to talk about. You guys notice how the axon terminal of this guy look like this bulbous thing? Why does the axon terminal of the postganglionic sympathetic neuron look like this? What is this called? Well, it's called a varicosity. Varicosity. And this varicosity is super useful for covering a huge surface area. Think about what the sympathetic nervous system innervates. It can innervate the cardiac muscle, the smooth muscle, certain glands, okay, and smooth muscle in the airways, smooth muscle in the vessels, right? So it requires such a high surface area that it has this sort of varicosity morphology, okay? All right, so what are these neurotransmitters that get released? What are these sons of guns going to be called? They're going to be called, and you know, if you're saying norepinephrine and epinephrine, you are wrong. It's only going to be norepinephrine. Where do we get epinephrine from, guys? Epinephrine comes from the adrenal medulla. I'm not going to draw it. I mean, I'll draw it like this. It's this triangular shaped looking thing, right? But those guys make both epi and norepi, okay? How? Via the enzyme PMNT. I believe it's phenylethylamine methyl N transferase, okay? That's the enzyme. And that they're actually specifically going to make 80% epinephrine and 20% norepinephrine, and they're going to secrete both of those directly into the bloodstream, okay? Directly into the bloodstream. All right, so remember that. That's the special scenario there. That's where we get epinephrine from, not from the nerves, okay? It's so important for you to understand this. You don't get epinephrine from the nerves. You get them from the adrenal medulla, okay? Special cells in here called chromaffin cells. All right, let's erase all this and go back to our original topic in the video. So norepinephrine gets released here, right? This is a special neurotransmitter. What is he going to bind to here? There's two receptors. There's two receptors here, okay? And, they have a special shape. We call this a serpentine receptor, okay? Or a 7-pass receptor because it actually passes through the membrane 7 times, okay? So it goes 1, 2, 3, 4, 5, 6, 7, okay? It does that. Now, these receptors, okay? These receptors, at the ends of them, they have a special thing. They have a G protein bound towards them, okay? A G protein. This is why you'll sometimes see these be called G protein coupled receptors or secondary messenger receptors, okay? Because this G protein is going to act as a secondary messenger. Okay, so this neurotransmitter, this norepinephrine, it's very specific to this receptor. And I'll tell you why in a second, okay? What is this receptor going to be? What is this receptor going to be? It's got a special name, guys. This is an adrenergic receptor, okay? Specifically in the heart. In this case, we're talking about the heart. So this is the beta 1 receptor, okay? This is classified as a metabotropic. Metabotropic. Why metabotropic? Because it has a G protein. Okay, it has a G protein. Anytime you see something that has a G protein, it's a metabotropic receptor. There are different types of metabotropic receptors. We can have the adrenergic ones, or we can have another one that is actually found on the heart. This orange guy. You know what this guy's called, guys? He's called a muscarinic receptor. Muscarinic receptor, specifically, since we're talking about the heart, M2, okay? It's going to be M2. You know what this guy is? What kind of receptor he is? He's also bound to a G protein, so it's a metabotropic. So you should kind of be seeing a pattern here already, right? Where we have different kinds of receptors. We had these ionotropic nicotinic receptors, okay, that bound acetylcholine, and we have the muscarinic metabotropic receptors, and we have these adrenergic metabotropic receptors. Now, adrenergic, why adrenergic? Any fiber that releases norepinephrine or epinephrine is going to be called an adrenergic fiber, okay? So an adrenergic fiber, okay, this guy, he's an adrenergic fiber, he's going to acquire an adrenergic receptor, okay? That means that this norepinephrine is not going to bind here. It can't. It literally can't. What can bind here? What can bind here? What can bind to this muscarinic receptor? Acetylcholine, guys, acetylcholine, okay? So again, this example here is going to be about the heart, okay? Heart. And you guys know that the sympathetic system increases heart rate. It increases the heart rate. But the parasympathetic system lowers heart rate. So if, for example, acetylcholine, right, if this wasn't a sympathetic neuron and this was a parasympathetic neuron, so let's imagine this. It's a parasympathetic neuron right here, okay? By the way, the parasympathetic neuron also has a varicosity. If the parasympathetic neuron released acetylcholine, that would bind to this muscarinic receptor, okay? It would bind to this muscarinic receptor, and it'll activate this G protein, okay, which will go do in other things, okay? It'll actually lead to your heart rate going down. This guy would eventually lead to your heart rate going up, okay? So that's that. Okay. Let's continue off with this. What does this G protein do? So the beta-1 adrenergic receptor bound its norepinephrine, okay, or noradrenaline, whichever you want to call it. And now this G protein is going to get released and go do a whole bunch of other stuff. One of the things that can happen, though, is that it'll activate a membrane channel. It'll open up a membrane channel, and this membrane channel is going to allow for the, oh my goodness, I'm trying to find the color here. It's going to allow for the leakage, or, for the entry of positive charges into the cell or negative charges out to the cell, okay, out of the cell. Why? What's the whole goal here? The entire goal is to depolarize this cell, okay, or is to make the inside of the cell a whole bunch more positive, okay, make it more positive. So how can we achieve that? We can either push out a bunch of negative ions, or we can bring in a whole bunch of positive ions. So we can maybe push out chloride, and we can maybe bring in sodium, okay? I want you to also remember that this, it doesn't happen like this, okay? So they both aren't coming in at the same time. It's one or the other, okay? So maybe this will come in, or maybe chloride will go out, okay? All right, so just making sure you guys understand. Okay, and again, negative and positive, boom. Great. So that's what's going to happen. Now, we usually call the metabotropic receptors an indirect pathway. Indirect pathway. Why indirect? Because it indirectly, guys, opened up an ion channel, okay? Whereas when we talk about ionotropic channels, we call these a direct pathway, okay? Why direct? Because they directly allow for ions to come through its own channel, okay? And again, the fact that I've put these receptors where they are is not by accident. This is exactly where they will be located. So there will be an ionotropic nicotinic receptor on the cell membrane of the postganglionic neuron, and there will be either a muscarinic or an adrenergic receptor. Well, actually not either, both would be here. And depending on which, neurotransmitter is released and binds to which receptor, then you will see the effector function, okay? So whether it's a decreased heart rate or a higher heart rate, okay? So that's what's kind of happening. All right, just to quickly recap everything very quickly, okay? We're zooming in on what's happening in the nervous system. Here's the spinal cord in pink. Here's the sympathetic chain ganglia, and we were looking at the interaction between this blue neuron and this purple neuron, okay? So what we did is we zoomed in on it, and we saw this blue neuron was getting stimulated spatiotemporal summation-wise, which led to a whole bunch of EPSPs summating on top of each other, generating an action potential, okay? How do you generate an action potential? You have to hit threshold potential. Once you hit the threshold potential, the sodium and potassium voltage-gated channels open, action potential propagates along, opens up eventually at the synaptic terminal, opens up these calcium channels, which come and allow for the binding of synaptotagmin to the snare proteins, which causes the exocytosis of these neurotransmitters. What kind of neurotransmitters get released here specifically? Acetylcholine gets released. What else? This acetylcholine is going to bind to its receptor, nicotinic acetylcholine receptor, which is also an ionotropic channel. That's going to allow for the opening of this channel and allowing sodium to come in and eventually depolarize this postganglionic cell. When it depolarizes the postganglionic cell, what's going to happen? You're going to reach threshold potential. You're going to do the exact same thing you did there. Launch an action potential, open up these guys at the axon hillock. Axon hillock. Remember, highest concentration of the voltage-gated channels. All right? Great. Eventually, what's going to happen? These positive charges that are coming in, they're going to stimulate the opening of this calcium voltage-gated channel. This calcium voltage-gated channel, which, by the way, opens at about negative 40 millivolts, is going to allow calcium to come in, and it's going to facilitate the binding of synaptotagmin to the snare proteins, which eventually causes exocytosis of the material inside. Okay, what are the material inside? Neurotransmitters. What kind of neurotransmitters are going to be released from the sympathetic nervous system postganglionic neuron? Norepinephrine. Remember, epinephrine only comes from the adrenal medulla, but norepinephrine comes from the postganglionic sympathetic fibers as well as the adrenal medulla. What is this norepinephrine going to do? Is it going to bind to the muscarinic receptors? No, it's not going to bind to the muscarinic receptors. It's going to bind to the adrenergic receptors, okay? Once it binds to the adrenergic receptor, it'll activate the G protein that's bound to it, causing it to dissociate or actually separate from this receptor, which will go do a whole bunch of things. One of them being activating a channel on the membrane, which allows for positive charges to come in, or it can allow for negative charges to go out. Either way, depolarizing the cell, which in this case would lead to an increased heart rate, okay? Now, just a couple of things to note. There are other types of adrenergic receptors. So there's alpha-1, beta-1, beta-2. There's all kinds. There's alpha-1, 2, 3. There's beta-3, 4. You don't have to worry about where these guys are located, but just know that they're located at tissue-specific regions, okay? So this means that each tissue contains a different set of adrenergic receptors. And the same goes for muscarinic receptors. Muscarinic receptors have 5 types, so M1, M2, M3, M4, M5, and they usually alternate between being stimulatory, inhibitory, and so on, okay? Just remember that. The ones I want you to remember are these: M1 and M2, stimulatory and inhibitory, okay? And again, it makes sense, right? We're talking about inhibitory receptor. This guy will eventually lead to lowering the heart rate, okay? Inhibiting the increase of heart rate. Okay. And like I said, they bind acetylcholine. What about the nicotinic receptors? There's not many types of them, okay? It's just this. He also binds acetylcholine. Okay. All right. Hopefully all that stuff made sense, guys, okay? That was a review of the nervous system, the big chunks of it, okay? And all the components of it. But now we're going to take a look at this graph here and how it correlates to this diagram we drew here. So, just to orient you, on the y-axis we're going to have the millivolts, and on the x-axis we're going to have time in milliseconds, okay? So, negative 70 is your resting membrane, negative 55 is your threshold potential, okay? Positive 30 is the peak of depolarization, okay? Peak of depolarization. Okay, I'm not gonna write this down. I need some more space later on, but you guys should know what that is. Okay. Now we're gonna look at the orientation of some of these channels at different parts of the action potential. So you guys already know that summation needs to occur to launch an action potential. So here's an example, right? Summating. Okay, it didn't take, but guess what? Something else is going to happen. Another inhibitory neuron will come in and he'll launch an IPSP. So this portion in green is an EPSP that is being summated. Okay, so it's summating EPSPs. And this is an IPSP. Okay, inhibitory postsynaptic potential. And then you get some more EPSPs, which can eventually make you reach threshold, okay? Why am I telling you this? Because when you want to launch an action potential, this is kind of how it looks like, okay? This is the setup that it looks like. It's not just one EPSP and boom, you hit action potential. It's multiple EPSPs summating on one another, maybe some IPSPs, and then maybe some more EPSPs. The combination of these guys, of the EPSPs and the IPSPs, the net change is going to be called the grand postsynaptic potential. Okay, so the grand postsynaptic potential. That's what that's going to be called. Okay. Just wanted to make sure you guys understand that crucial concept before we move into what all of these channels look like at rest. So here's that resting membrane potential, right? Let's start with number 1. What is the configuration of the sodium channel and the potassium voltage gated channel? Okay, So here's the sodium voltage gated channel, here's the potassium voltage gated channel. Again, it's all color coded with the above here, okay? So the sodium voltage gated channel will have an closed activation gate and an open inactivation gate, okay? And the potassium voltage gated channel, it will have a closed activation gate. Okay, cool. What is it going to look like as soon as we hit threshold potential, so negative 55? I'm so glad you asked. This guy, this activation gate, is going to snap open very fast. But this guy, this inactivation gate, is going to slowly close, okay? This right here, this arrow that I'm putting, this means slowly closing. And it takes about 0.5 milliseconds for this guy to close, okay? That's going to be important, remember that. This guy, okay, what about this guy? He's slowly opening, okay? Very, very slowly opening, okay? So I'm going to write down slow open. But did you notice all gates open up at, or start moving at negative 55, okay? All of them. Remember that. They all start moving at negative 55. It's just they have different speeds. Okay, so this one is fast. This one is slow close. This one is slow open. Okay. So what happens when we hit the peak of depolarization? The peak of depolarization, what's going to happen? This guy is going to be closed. This guy, the inactivation gate is closed, while the activation gate is fully open. Okay, what about the potassium channel? Well, the potassium channel, the activation gate is now fully open. It's now fully open, okay, and now it's going to allow for potassium to actually, sorry about the other way around, potassium to actually leave the cell. So potassium leaving the cell, okay, which makes the inside more negative. Now, potassium is going to continue to leave the cell until down here. Okay, it's going to cause a hyperpolarization event. So this portion is repolarized, is repolarization, okay, from here to about here. And then this portion to here is hyperpolarization, okay? And then, of course, here to here was going to be depolarization, okay? You guys already know that. Just want to make sure you guys know this stuff, okay? Great. So, like I said, potassium is going to continue to leave, okay? And why is that the case? The case is it's going to continue leaving that because this activation gate, not only was it slow at opening, but it's also slow at closing. So I'm going to put here this arrow again that I told you means slow. It's a slow closer. It closes very slowly, okay? It takes a while to close. That's why we get a hyperpolarization. We had a whole bunch of extra potassiums leave the cell, okay, which hyperpolarized the cell, made it more negative than it actually was. It brought us to maybe about negative 80, okay? What was the configuration of this guy? Students sometimes get confused here, but as soon as we hit the resting membrane potential, the configuration of the sodium voltage channel goes from this immediately back to what it was at rest, where the inactivation gate is open and the activation gate is closed, okay? Great. And if you take a look at number 5, I put an arrow saying that it's the same as number 1, okay? That's why I'm not going to cover it. All right. Now, a couple more things, guys, and we'll be done, okay? We'll be done. This portion here of the graph is called the absolute refractory, okay? This portion of the graph is called relative refractory. And by the way, the entirety of the action potential takes about 1 millisecond. Remember I told you remember this number? It's going to come in handy. I'll tell you why right now. I told you that this inactivation gate takes about 0.5 milliseconds to close. If the entire action potential, so from here to here, is 1 millisecond, 0.5 is going to be about half. So it's going to be here. Would you look at that? At this halfway mark, this channel is closed, okay? And if this channel is closed, we can't go higher than positive 30, okay? And incidentally, this channel opens at that time, so we actually start going back down, Okay? But now, this is the reason why they call it the absolute refractory, because of the configuration of this sodium voltage gated channel at positive 30. Because the inactivation gate is closed. Okay? This is called the absolute refractory. What is the absolute refractory? This just means that we can't launch another action potential at this point. We can't. It's literally physically impossible. Why is it impossible? Because of the configuration of this channel. Okay? Because this inactivation gate is closed, we can't launch another action potential. Okay? But you're probably saying, hey, Keon, you said absolute refractory, you can't launch another action potential. But what about relative refractory? Well, in the relative refractory, you actually can launch an action potential. And tell me why, guys. Tell me why. If you're telling me that it's because of the configuration of the sodium voltage-gated channel, you would be correct. Because look, it's the same as it was here in the start. The inactivation gate is open while the activation gate is closed. Okay, so we can launch another action potential here. It's just going to be much harder. So the stimulus that got us to action potential here might not reach action potential here. Okay? We need to have a way more stronger stimulus to kind of get us to threshold here. And that should make sense to you because the cell is more negative at this point than it was at resting membrane. Okay? All right. So that's the absolute refractory, relative refractory, all of the channels and what they look like, and you know, the actual kinetics of the channels. Okay? All right, guys. That's going to finish it for this video. Again, if you liked it, if you learned something out of it, and you enjoyed it, please comment, like, subscribe. If you have any questions about it, comment. I'll try my best to answer. But that's going to be it for today, guys. Thank you very much.